Hello everyone. Let's begin with today's session on Enzyme Sciences and Technology. So in today's class, we will study mostly about enzyme assays and enzyme kinetics. So in the previous class, we had studied various enzyme catalyzed reaction and how do we measure these enzymes assays by different techniques like chromatography, spectroscopy. So today also we will study about enzyme assay, and there will be a few techniques which are repeated like spectroscopy and electrophoresis, and finally we'll move on to enzyme kinetics. So to begin enzyme assay, so enzyme assay, as you know, we have in any assay we have a enzyme and substrate. Okay, so we will have enzyme which will react with the substrate molecules, and it will give us enzyme substrate complex ES. So this complex can be further converted into enzyme product. So this enzyme will convert substrate into enzyme product complex, and then from this enzyme product complex we'll get enzyme plus product, and this free enzyme is what we get in the end can further convert other substrate molecules into product, and the reaction will go on. So this is the general scheme of enzyme substrate interaction, and so in order to measure, so by enzyme assay we can measure different moieties, like we can measure substrate and we can measure product concentration. So the substrate here is getting utilized, that is, it is getting converted into product. So we can measure substrate depletion, and also we can measure product formation. So enzyme assay could measure either of these two things. Either we could measure substrate depletion or substrate getting utilized, or we can measure product formation. The product is formed over a certain period of time. Okay. Over a certain time. Then there are other, in order to plan enzyme assay, we need to consider few things, few considerations need to be taken. First is the cost of enzyme assay. The cost of enzyme assay shouldn't be high, it should be economical. Any assay, if we are planning, it should be economical, and the cost shouldn't be high. Next is whatever equipment or technique which we are using, that equipment of choice should be available, should be easily available. The equipment or the technique which we are doing, it should not be costly, and the equipment also should not be rarely present. So equipment also comes in the cost. Then the next consideration is that we need to measure true molecules or true activity, as in it should be measuring true activity is one of the consideration. So measuring true activity or of enzyme. So by this we mean that there should not be any contamination or other side reactions that we are measuring, or other color formation of or the substrate getting uh, converted to something else to other products, so there should not be any contamination of other molecules. We should measure the true activity of enzyme, and we should also plan adequate controls in our experiment. So the controls are very important in any enzyme assay. They decide the actual they actually decide the measure the true activity of any enzyme. So controls are very important, and we need to have proper control, positive control, negative control, and also uh, blanks uh, which are required. So these are the considerations which we need to keep in mind in order to plan any enzyme assay. So next we move on to how the enzyme assay behaves. So as we know that from our enzyme substrate interaction, so we have enzyme giving us enzyme substrate, and this gets converted into enzyme plus product. So this complex, so it is very difficult to measure enzyme substrate complex. So we have a product formation here, so on the y-axis, and as you can see, as the time proceeds, there we have a log phase here, so we have a logarithmic phase here, and after certain time, like after 20 or so, there is not sharp increase in the product formation, there is a saturation. Okay, so we see a saturation here, and this saturation could be because of many reasons. Okay, so this saturation or loss of linearity, this saturation or loss of linearity could be of because of many reasons, like the substrate has depleted. Whatever substrate which we had provided in the assay, that has got depleted. This could be one of the reasons. So that is why we are not getting same amount of, or we are not getting a logarithmic increase in the product formation, because the substrate has depleted. If there is no substrate remaining, there won't be any enzyme substrate complex, and there won't be any product formation. So the substrate got depleted, so that's why we get this saturation or loss in the linearity. The another reason could be that the equilibrium has reached. So what is this equilibrium? This equilibrium is between formation of this enzyme substrate complex and uh, uh, and the free enzyme and free substrate. So this could be because of many reasons, that if the equilibrium has reached, it could be because that the rate of both formation and degradation of enzyme and substrate could be same. That is why it has reached a equilibrium. So we need to consider the rates also in mind, and not allow these reaction to go into equilibrium. Then there could be product inhibition. So whatever product is getting formed here, okay, it is getting inhibited by some of the molecules present in the assay. Okay, and this molecule could be any irreversible inhibitors or reversible inhibitors. These molecules could be any contamination from the assay. Suppose we have a cell lysate, so there could be different inhibitors present in the lysate, which could have a irreversible inhibition on the product itself, and that's why the product formation has decreased, or it has reached a saturation. Another reason could be that maybe the assay is time dependent. By this I mean that the assay will stick to particular time. Suppose a enzyme reacts only for 30 minutes, and after that all the resources or all the conditions are not satisfied for the enzyme assay, and thus there is the less generation of product, and thus there is loss in the linearity. That's why we are not getting constantly, there is no constant product formation. Okay, see. And another reason could be that the product which is formed is unstable. The product is unstable. So the product might be unstable in the assay and it is getting degraded. So whatever product we are measuring, that is not that is undegraded product, and the product which we are getting, suppose we are getting 100 molecules of product, around 50 molecules might be getting degraded because of some assay conditions. Okay, so these 50 molecules which are getting degraded are not measured by our enzyme assay, and thus we are getting thus there is a loss in the linearity. Okay, so these are the reasons in which the enzyme assay might behave in might reach saturation, or there will be a loss in the linearity. Okay, um, so the solution could be, solution of all this could be that we could measure the initial rates only. That is, we could measure by initial rate, that I mean only till this time. Okay, let's say suppose these are 15 minutes, so we can measure only initial rates. That is, we can plan assay that would measure only the first 15 minutes of the overall assay conditions. So, and this measuring initial rates has many advantages. Okay, it has many advantages. That is, during this linear phase, okay, this linear logarithmic phase, the enzyme substrate, the enzyme assay is very sensitive. Enzyme assay is very sensitive. That's why it is better to measure only the initial state. And the reversible and the equilibrium doesn't reach. Thus, the reversible reaction, that is the formation of free enzyme and substrate, will not occur. So, at the initial rates, the formation of ES is pretty high as compared to this backward reaction. So, if this is K1 and this is K minus 1, the formation of the K1 is higher as compared to that of K minus 1, and because of that there will be formation of enzyme substrate complex, and this backward reaction is inhibited, or there is not an equilibrium observed here. So that's why we measure reaction at initial conditions. Now, blanks are also important while measuring the enzyme assay. So what is this blank subtraction? So suppose we have two tubes with us. In one tube we add our buffer or any solution for the assay. So this is tube A and tube B. So we add we add substrate and buffer here in A. That is, A is without enzyme. So A tube is without enzyme and B is with enzyme. So in B we add substrate plus enzyme plus buffer. So without enzyme there won't be any reaction, or there won't be any much reaction observed. So we'll measure the absorbance of unreacted substrate. We'll measure absorbance of the unreacted substrate, and let's say we get some absorbance like 0.1. So this is the minimum absorbance that we are getting, and this absorbance is just because of the buffer solution or the substrate molecules present. Okay, so this absorbance 0.1 is supposed to be this 0.1 absorbance is supposed to be subtracted from the overall final absorbance, because this absorbance is actually because of the substrate or buffer present in the tube. And with the enzyme we are getting there would be enzyme substrate reaction, there will be formation of some product molecule, and that product is giving us a higher absorbance, so the absorbance of the product, and we are getting some higher absorbance like 1.1. So this 1.1 is absorbance is because of the enzyme substrate reaction, and this reaction is giving us some product, and that product is giving us a higher absorbance. So when we finally measure our absorbance, we'll subtract the blank, that is 0.1, we'll subtract it with our, so this is our blank absorbance, so we'll subtract it with our reaction absorbance. Okay, we'll subtract it with our reaction absorbance, that is 1.1, and our actual absorbance are the actual absorbance of the enzyme substrate reaction of the or of the product is 1. Okay, so this is necessary to have the actual absorbance. And why is this necessary? So if you don't subtract subtract the absorbance, we'll get wrong readings, we'll get a higher reading. So this is, suppose this is a normal linear reaction of product formation with time. Okay, we are measuring the initial rates, so this is the subtracted absorbance at different time points. Okay, subtracted absorbance, A is the subtracted absorbance. If we don't subtract, then we'll have a graph looking like this without any subtraction, and this graph will essentially give us wrong readings, because this graph will have a blank or negative absorbance as well as in this, and it will not give us true readings or true activity of the enzyme. So that is why it is very important to have a blank subtraction. Okay, and this reduces the error also. It reduces the error which we are getting from the assay mixture, and also reduces any contamination. Okay, so this is why it is very important to have a blank subtraction. Then so we get this different types of graphs. So we have a upward and downward curvature graph. So what these graphs say? So this upward curvature graph here, what does it say? So this is because of the contamination which are present in the, so this is the normal straight line graph. This is how it is supposed to look. But in the B graph, as you can see, there is a delay at the initial time points, okay, at the initial enzyme concentration, and then it is increasing linearly. Okay, now this delay is due to irreversible inhibitor present in the assay mixture, okay, which is actually inhibiting the enzyme at the initial condition. It is inhibiting some fraction of the enzyme, and after that there is a linearly increasing it, the reaction rate, the velocity is linearly increasing. Okay, so this B is because of the presence of irreversible inhibitor. So whenever there is a upward curvature, it is because of a irreversible inhibitor present, and this C is because of a reversible inhibitor. So whenever a reversible inhibitor is present, that is, the inhibitor is reversibly binding to the enzyme. Okay, so we have a smooth curve smooth upward curvature in case of smooth upward curvature in case of reversible inhibitors. So these are the special conditions which can be seen during your enzyme assay, and you need to recognize it is why is it because what is the reason of these upward and downward curvature. And whenever there is downward curvature like this, the downward curvature is mostly because of depletion in, again, the reasons which we mentioned previously, depletion in the substrate concentration, or the substrate is getting degraded, and because of that the product formation is also, the velocity will also reduce. So there are both upward and downward curvature. So downward curvature I had already explained, like what are the reasons to get downward curvature, and this upward curvature are mostly due to irreversible and reversible inhibitors. Now we'll measure the enzyme activity. So in order to measure enzyme activity, first we need to have enzyme activity units. Okay, enzyme activity units, and it is very important to have this enzyme activity units in order to correctly determine its activity. So enzyme activity unit is defined by so we have a unit measurements, that is where 1 unit is equal to 1 micromole of substrate getting converted to product, in 1 minute. This is very important. So 1 unit refers to 1 micromole of substrate getting converted to product in 1 minute. Okay, so 1 unit is equal to 1 micromole of product formed, in 1 minute. Okay, so this is the definition of units. So if I say 3 enzyme unit, it means it is converting 3 micromole of product in 1 minute. Okay, it is converting 3 micromole of product in 1 minute. Then we have specific activity. So this specific activity was created in order to calculate the efficiency, or how much enzyme is calculate the specific activities, because mostly is done due to calculate the efficiency, and it considers the amount of enzyme. So the specific activity refers to 1 unit per milligram of enzyme, of the enzyme. So again it will become specific unit will become 1 micromole 1 minute into 1 mg. Okay, so it considers the amount of enzyme present. Okay, it also considers the amount of enzyme present, but sometimes an enzyme can have multiple binding sites, multiple active sites, so we have a different measurements known as catalytic center activity. And what this catalytic center activity refers to, it refers to, instead of 1 mg we have number of active sites. So it will give us so the formula is 1 unit upon number of active sites. So again this will become 1 micromole upon 1 minute into number of active cells. Then these are the traditional methods, traditional terms used to measure enzyme activity, and recently we use kettle. So what is this 1 kettle? 1 kettle is equal to, in terms of unit, it is equal to 60 mole. 60 mole. 1 kettle is 60 mole per minute, and in terms of unit it refers to 6 into 10 raised to power 7 units. Okay, so 1 unit, so we can convert this also. 1 unit in terms of kettle, we can write 1 unit is 1 micromole per minute, so it will become 16.67 nanocatals. Okay, so this is the conversion between units and kettle. Okay, so this many people use either kettle or 1 unit. And so while measuring enzyme activity, we need to also consider temperature, pH. We should consider temperature, pH, and assay conditions, like what all cofactors or coenzymes are required. These are also considered during enzyme activity measurements. Okay, next we'll move on to different types of enzyme assays. So enzyme assays are mostly of can be divided into 4 types. Radiometric assay, which uses radioactive isotopes. Then we have photometric assay, in which we measure absorbance. We measure fluorescence, and we also measure luminescence. So all light related assays comes under photometric assay. And in chromatographic assay, as we already have studied chromatographic assay and gel electrophoresis assay, so I won't be going in this in detail in this class about chromatography and gel electrophoresis, but we'll study more about photometric and radiometric assay. So let's begin with photometric assay. So so in the photometric assay, as I mentioned earlier, so the photometric assay considers it involves light or optical properties of either product. If you want to measure product, it could be optical activities of the product, or it could be optical activities of the substrate. It could be either one. So there are many advantages of photometric assay. It's widely used because it's very easy, it's cheap. We have different spectrophotometers present in our lab, so you can easily measure by using different absorbance of fluorometers or spectrophotometers. It is very cheap, economic as well. Okay, the reagents are also very cheap, and the photometric assay could be either of indirect measurements or it could be direct measurements. So what is indirect assay and direct assay? So indirect assay is nothing but so let's first understand what is direct assay. So in direct assay, we are directly measuring, as the name suggests, we are directly measuring either product or substrate. So we are directly measuring. So suppose, let's say if we are getting, so we have a glucose, which is getting converted to glucose 6 phosphate, okay, by enzyme hexokinase, right? So this glucose 6 phosphate, we can directly measure this. Suppose this glucose 6 phosphate is yellow in color, so we can directly do a absorption spec. Okay, we can measure this yellow color by change in absorbance. Okay, so this is a direct measurements. Okay, and what is indirect measurements? In the same assay, in the same glucose to glucose 6 phosphate, we also have ATP involved. Okay, and we have here ADP, we get ADP. So we can measure in indirect assay, we can measure this ATP. Okay, so we are not directly measuring the substrate or the protein here, okay, but we are measuring the side product or the end product, or it is getting converted to something. So we can measure, either measure the depletion of ATP, the depletion of ATP, or we can convert this glucose 6 phosphate into some other chromogenic substrate. Okay, we can convert this glucose 6 phosphate into some other compound. Okay, and that compound is we can measure by either different spectrophotometric assay. Okay, so in indirect method, we are converting our substrate or product into a different compound, and we are measuring that. Okay, so in direct, both indirect and direct assays are, they could be either they could be either continuous measurement, or the both indirect and direct assay, they could either be continuous or discontinuous measurement. So what do you mean by continuous? So suppose I have so by continuous measurement I mean I add so we have different time measurements here, and we have velocity or product formation. Okay, either of two. Okay, velocity or product on the y-axis. So we are continuously measuring with respect to time, we are continuously measuring the product formation. Okay, so it's a time dependent, continuous is a time dependent, we are continuously measuring the time up to how much ever time we want. So that is a continuous measurement. And what is discontinuous? In discontinuous, we have a end product, we have a formation of a end product. Let's say we have a product form at this time. So we'll stop this reaction, after it reaches this product. We'll stop the reaction by adding some reagent. Okay, by adding some stopping reagent, we'll stop the reaction, and we'll measure the product at different time points. So let's say we stop here. Okay, so this is P1 at T1, and if we the next reaction we can stop at P2, next reaction we can stop at P3. So this P1, P2, P3 is also with time, but we are stopping the reaction, and we are measuring how much product is formed at the end of every time. Okay, at the end of every time interval, as you can see here. So this is the discontinuous method of assay. So coming back to photometric assay, so we have a xenon lamp here. So this photometric assay is nothing but measuring the fluorescence. Okay, so what is this fluorescence? Whenever light is getting whenever we have emitted light. Okay, so if this molecule, it will absorb this light, it will go to the excited state. Okay, and it will come back to the ground state. Okay, while coming back to the ground state, it will release some energy in form of light, and that light is known as emitted light. Okay, and this is the emitted light. Okay, so this is the cuvette, Okay, and after getting excited from the xenon lamp light, so this is the absorbent, this is the excitation light. Okay, this is the excitation light. After getting excited, it will release the emitted light. Okay, this is the emitted light, and this emitted light is collected by the emission monochromator and it is detected. Okay, it is detected. So this is how, this is a fluorometer. This is the representation of a fluorometer. Um, so the fluorometers could be of, so we are measuring the emitted light at 90 degree. Okay, so this is not a absorption spectroscopy, this is fluorescence spectroscopy. The light traveling could is emitted in the all directions, so we are just measuring the emitted light at 90 degree. Okay, so the examples for this is, so similarly we can measure luminescence as well. So let's take one example. Suppose we have a luciferase enzyme which is getting, we'll take the same example, that glucose plus ATP will give us glucose 6 phosphate. Okay, and then we have, to this we'll add a luciferase enzyme. Okay, to this we'll add a luciferase enzyme in presence of O2. So this luciferase will give us oxyluciferase. Okay, and this will, and it will it will show a luminescence. Okay, it will show luminescence. Okay, and this luminescence will be measured on a luminometer. This luminometer is same as that of the fluorometer. Okay, and so so this is a discontinuous measurement. We stop this reaction by adding H2O2. Okay, we stop this reaction by adding H2O2, and by adding H2O2 we'll get with the luciferase here, it will interact with the ATP molecule, whatever ATP molecule, and we'll get a reading, we'll get a luminescence here. Okay, and this is the example of one of the luminometer or luminescence. So some photometric assay could be coupled also. So there could be a coupled assay. So what is this coupled assay? So in the same coupled assay, let's say from glucose plus ATP to glucose 6 phosphate, the same reaction. This glucose 6 phosphate can be coupled to a different reaction where this could be coupled to a dehydrogenase reaction, where this glucose 6 phosphate, okay, the product is converting NADP plus, it is getting converted to NADH plus. Okay, and this NADH plus we could measure by UV visible spectrophotometry. Okay, so this is the example of a coupled assay, that we are not measuring glucose 6 phosphate but we are measuring NADH plus, and this NADH plus is getting converted to this NADH is getting formed because of glucose 6 phosphate, which is our product in the reaction. So so this is another example of a continuous coupled assay. Okay, this is another example of a continuous coupled assay. Apart from photometric assay, we also have turbidometric assay. So instead of measuring absorbance in a turbidometer, we measure the turbidity. So what is this turbidity? It measures the scattering property. It measures the scattered light. Okay, so in fluorescence we measure the emitted light. In turbidometer we measure the scattered light. So how do we get this scattered light? For example, if we have enzyme, so let's take an example. If we have a lysozyme, if we are measuring, doing the assay for lysozyme enzyme, so this lysozyme breaks open the cell wall. It breaks bacterial cells, okay, and it releases the, and it breaks open and it releases the content, which increases the turbidity. Okay, and thus if we can measure lysozyme by measuring the turbidity. So suppose so we have cells, intact cells, will measure the absorbance or the scattered light. Okay, it will give us some scattered this will be our blank. Then in the test we'll add our cells plus lysozyme. Okay, and then we'll measure the absorbance, and the absorbance will increase. Okay, the absorbance will increase. Okay, there will be a relative increase in the absorbance. So so this is also a type of a photometric assay where we are measuring the scattering of lights. Okay, now let's move on to radiometric assay. So in the radiometric assay, as you can see, we measure different radio labeled isotopes like hydrogen 3H, 14 carbon, 32 P32, P33, and 35 S35 sulfur. Okay, like that. So we measure different radioisotopes, and this radioisotopes or radiometric assay could be coupled with different techniques like chromatography. It could be coupled to chromatography, and it could also be coupled to TLC or other techniques. Okay, so in the radiometric assay, basically we are using the radioisotopes, and then we so one of this radiometric assay is scintillation proximity assay, also known as SPA. So this SPA could be immobilized onto a bead. Okay, so this SPA can be immobilized onto the bead, and this SPA will have a radioactive particle. Okay, that particle will emit light in presence of autoradiography, in presence of any radiography, in presence of any it will radiate radiation. Okay, basically. So and that process is known as autoradiography because it itself is emitting a radiation. Okay, and so this is how you can couple the SPA assay to different chromatography techniques. So these beads could be biotin label also, and then you can use a biotin-ylated tag and do a SPA assay. So these are the different examples of different enzymes that can be assayed by SPA techniques. So we have here different enzymes: HIV integrase, telomerase. So the telomere here could telomerase enzyme has affinity for telomeres of DNA. So we could have DNA bound to the beads, or DNA bound to the beads, different types of beads, and then we could do SPA to similarly we have DNA helicase, RNA helicase, and RNase H. So this could be these enzymes could couple with different chromatographic techniques, okay, in order to do different assays. So let's move on to enzyme kinetics. So enzyme kinetics uses two approaches. So enzyme kinetics is basically measuring of enzyme, how the enzyme is evolving over the period of time. Okay, how the enzyme rates are evolving. And why do we need kinetics for that? Because measuring enzyme substrate complex, that is ES complex, is very difficult. Measuring this enzyme substrate complex E plus S gives us ES. To measure this enzyme substrate complex is very difficult. So we need to have a kinetic parameters with us in order to find out how much how the kinetics are evolving over the period of time. Okay. So we have two approaches here. Rapid equilibrium approaches by Henry Michaelis Menten. Okay, in this approach, what happens? The early component of the reactions are at equilibrium, that is E plus S. Uh, uh, so these early component, that is E plus S, gives us ES. So these are at equilibrium. Okay, so this permits one to express ES in terms of so here this Ks is the formation of enzyme substrate complex, and this is K minus 1. Okay, the backward reaction is K minus 1. Okay, so let's find out how to find out the velocity for this reaction. Okay, so by using the first approach of Henry Michaelis and Menten. So we have enzyme plus substrate giving us enzyme substrate, where this is Ks and this is K1. Let's say this is K1, forward reaction, and this is giving us K2 E plus P. This is giving a. So measuring this enzyme substrate complex is very crucial. Okay, so it's very crucial, and following the first assumption, the velocity of the formation of product, is directly proportional to the enzyme substance. So complex. So if there is a large amount of enzyme substrate is formed, it will get converted to product because there is no backward reaction here. So enzyme substrate complex, so product formation is directly proportional to the enzyme substrate complex. Okay, so if we remove this proportionality, we'll add a rate constant. Okay, so this is our first reaction. Okay, now the total enzyme ET is free enzyme plus bound enzyme, that is bound with the substrate. Okay, let's say this is our second equation. Okay, so if we divide equation 1 and 2, what we'll get? We'll get V upon ET. Okay, if we divide this and this, we'll get V upon ET K2 ES E plus ES, right? So also we know that Kd, or dissociation constant, let's say Ks, is, so enzyme can be dissociated into so this is the dissociation, that the enzyme substrate complex is dissociated into E and S. Okay, and its rate is K minus 1. Okay, so we can substitute this value in this equation, and finally we'll get V upon ET will give us K2 Okay, and if we solve this, we'll get V upon V max, substrate concentration upon Ks upon 1 plus substrate concentration upon Ks. Okay, and if we solve this further, we'll get V upon V max. If we divide this by Ks, we'll get okay. So what this means is that we have so this is the concentration of either on y-axis we have concentration, on x-axis we have time. So this is how the product graph will look like. Okay, it will reach a saturation. Similarly, at this point we'll have a highest substrate and the substrate concentration will lower. Okay, and this is how the substrate concentration will look like. Okay, and at this point the substrate concentration will be equal to product concentration. Okay, this point both the substrate and product will be equal. At the initial state, okay, at the initial state we have product getting converted to, we have very small fraction of enzyme substrate complex formation. Okay, so we are measuring this at initial conditions where there is a enzyme substrate complex formation. Okay, and here the enzyme concentration is decreasing. So this is the enzyme, the depletion of the enzyme. Okay, so this is the initial measurements. Okay, and so the next is the steady state, that is Briggs-Haldane approach, which is most widely used. So in the Briggs-Haldane approach, again we have enzyme plus substrate giving us enzyme substrate complex, and then we have E plus P, we have a product formation. This is K2, K minus 1, and this is K1. So this approach is also known as steady state approach. And it uses steady state hypothesis, that at steady state what will happen. So at steady state, rate of formation of ES formation, at steady state ES formation will be equal to ES breakdown. Okay, both is same, the rate of formation and breakdown is same. So again the velocity will be equal to K2 of ES, the initial velocity V0 will be equal to K2 ES, will be directly proportional to the this remains same, and we have enzyme total enzyme equal to enzyme plus bound enzyme and unbound enzyme. So if we write the rates of formation, so what are the formation of K1? So enzyme is formed by E plus S. So and the rate is K1. Okay, K1. This is the formation. This is K minus 1. Okay, and this is the formation, ES formation. And what is the ES breakdown rate? It is equal to K1 plus K2 ES. Okay, and if we simplify this, we'll get K1 upon K minus 1 plus, so this is minus 1, K2 enzyme and substrate, and this term this term is the Michaelis constant Km. Okay, and if we solve this, we'll get V0 upon V max, which is equal to substrate Km. Okay, so this Km is very important. Km is equal to substrate concentration when Km is equal to substrate concentration, then V0 will be equal to So Km is, when V0 will be equal to half of Vmax. Okay, so this is the condition. By this, what do we mean by this? So we have a V0 on y-axis here, and then we have substrate concentration. So this is our Vmax. So when when V0 is half of Vmax, the substrate concentration here, this is the substrate concentration at this point will be our Km. Okay, so Km is also known as Michaelis-Menten. Okay, and it also refers to the specificity of enzyme. It refers to the specificity of enzyme. Okay, then we have turnover number. Okay, what is this turnover number? Kcat. And Kcat refers to the number of substrate molecules converted to product per unit time. This is just like calculating the efficiency, that is Vmax upon Et. Okay, number of product converted will be directly proportional to the maximum velocity at given unit at Et. Okay, so instead of having so it is very difficult to plot a sigmoidal hyperbolic curve for enzyme kinetics. So in order to convert this into a linear plot, we can have a Lineweaver-Burk plot. So Lineweaver-Burk plot is nothing but if we take the reciprocal of Michaelis-Menten equation, we'll have a linear equation, okay, like this, and this linear equation can be fitted and have a linear graph with the intercept, okay, with the intercept 1 by Vmax, and the slope will give us Kn by Vmax. Okay, and this plot is known as Lineweaver-Burk plot. It is very useful in determining in study enzyme kinetics. It gives us easy measurements. So let's move on to assignments. This is the first assignment question. The enzyme activity can be determined in enzyme catalyzed reactions by measuring the enzyme concentration, observing the change in pH, monitoring the rate of product formation or substrate consumption, analyzing the reaction kinetics. So the correct answer is that we can monitor the rate of product formation or substrate. So the enzyme catalyzed reaction, we could either measure the formation of product or we could measure the depletion of the substrate. Okay, so either way it will give us the enzyme activity, that is velocity, or okay, it will so on this y-axis we have concentration, so it will give us the activity. So activity is nothing but either product form per unit time, right, or substrate utilized per unit time. So that is the rate of product formation or substrate consumption. It is recommended to limit the initial rate measurement to specific period of substrate consumption in order to initial rate measurements, we do initial rate measurements for multiple reasons. One of the reason is that in order to maintain the linearity, okay, so we do initial measurements. As you know, we have a initial linear phase and then we have a saturated log phase. So we do initial measurements, okay, at this is the initial measurements. It allows us to maintain the linearity. We have sensitive operations, okay, occurring in the linear phase. So that's why maintaining the linearity is So in order to maintain the linearity, we measure at initial rate. Okay. Blank rates in enzyme assay are rates observed at the beginning of the reaction, rates observed at the end of the reaction, apparent rate observed with complete assay mixture, apparent rate observed in absence of one of the assay component. Apparent rates apparent rate observed in the absence of the one of the assay component, and mostly that one of the assay component could be either enzyme or substrate. Okay, it could be either enzyme or substrate depending upon the control which we are using. Okay, the correct answer is D. The graph typically represented when plotting initial velocity against total enzyme concentration is, so how the graph will look like? So initial velocity and total enzyme concentration ET. The graph is supposed to look like a straight line passing through origin. Why? Because at total enzyme, so this is initial velocity always refers to the maximum velocity. So initially the graph will always be linear. Okay, that's why we do initial measurements in order to have a linear straight line passing through the origin. The scenarios that can lead to upward curvature in the relationship between initial velocity and enzyme concentration. So upward curvature is mostly because of irreversible inhibitors. Okay, so presence of irreversible inhibitors is responsible for the upward curvature. The main advantage of continuous indirect assay over discontinuous is that we can actually track the progress curves in a single assay. We don't have to do multiple assays. Okay, so the correct answer is D, that we can allow determination of the assay in a single. The relationship between absorbance and molar extinction coefficient when path length of curvet is 1 centimeter. So as per the Beer-Lambert's law, absorbance is directly proportional to concentration and path length. So if we remove this proportionality sign, we introduce this absorption coefficient. Okay, and if the path length is L is 1, this will be equal to epsilon c. So the correct answer is A. Scintillation proximity assay is a type of radiometric assay. So we use radioactive probes or radio-labeled molecules, isotopes. The signal decrease in scintillation proximity assay is most appropriate for hydrolases, transferases, polymerases, kinases. So the signal will decrease whenever because of hydrolases. Okay, in the SPA assay. The unit of absorbance is, so absorbance is basically log of I0 upon I. Okay, where I0 is the intensity. Okay, passing through reference upon intensity, that is, so basically it's a ratio of intensity, and that's why it has no units. In assay method describe the diamine oxidase. The fluorophore used is phospholipase, chitinase, elastase, homovanillic acid. Correct answer is D, homovanillic acid. The phenomena that contributes to non-linearity in fluorescence-based assay at high absorbance value is photochemical reaction, the inner filter effect, quenching effect, radiation-less transfer. The correct answer is the inner filter effect, B. In protein kinase assay, the peptide in the experimental condition migrates towards anode because, so the anode is positively charged. Okay, it is migrating towards a positively charged anode. Why? Because the peptide has a net negative charge. Okay, the correct answer is, that's why it is migrating towards positively charged anode. Dehydrogenase can be visualized using tetrazolium salt by Detecting changes in color of pyridine nucleotides, observing fluorescence changes in enzyme substrate reaction utilizing tetrazolium salt that becomes colorless upon reduction. Visualization of the oxidation without any color. The correct answer is C. The tetrazolium salt, utilizing the tetrazolium salt that becomes colored upon reduction. So, upon reduction, that is NADH conversion, it will give us a colored compound. The compound that is recommended as a substrate for peroxidase due to its non-carcinogenic property is 3,3'5'5' tetramethyl benzidine. Correct answer is C. So that's it. Thank you.